just say that I think.
Sorry.
Oops.
Yeah,
Yeah.
sorry.
I will move to full screen, so I probably won't be able to see the chat or anything. So if you would have any questions, can you just ask instead of putting the chat or okay, so let me start with that. How do I do this? I go away. All right, so thank you for the invitation. The talk today it's going to be about some results that my graduate student, Peg Melody, and I got pretty recently. I mean, this appeared in a paper last year in J Neuroscience, and I'm trying to kind of advertise as a method because I think it works beyond intracranial EEG recordings, can be applied as long as the signal is continuous. I guess LFP, EEG data, MEG data will work just fine. So, and it's about discovering attractor type of dynamics or separating different kind of trajectories. So I think one of the example I pick here is when the blue trajectory represents one percept and the red trajectory, it's actually representing a different percept. So that's something that we extracted from the patient, from the data of one of our patients. So first I would like to acknowledge, because I'm always kind of late in my presentation. So the main contributor was my graduate student, Peg Melland, who's right now at the Oregon Institute of Technology. And I need to acknowledge the Human Brain Research Lab, because the data was collected in this lab and they've done an amazing job. Usually the theory can go as much as the data is clean and nicely collected, I would say, if we work with real data. And especially Kirill Norsky was a collaborator in a previous paper talking about this kind of data. So what is all about? You may or may not be familiar, but a famous experiment which deals with bistable perception is binocular rivalry, when you show to participants wearing special glasses, two overimposed images, and because of the contradiction and the fact that each part of the glasses are picking up filtering one of the two images, there is this contradiction of terms, or this conflict, and the patient start to report one percept at a time, and then there's a switch to another percept, and then continues the switch back to the previous percept, and that's what you get, the so-called alternation. And that's an interesting experiment to run and to look at the data, behavioral data mainly, but obviously people try to correlate the behavior also with neural activity. In this talk, I will talk about a similar idea, but using an auditory percept. So basically, the stimulus was a sequence of triplets, and that's what I'm trying to describe here. You can imagine having two tones. I call them here A and B. There's a difference in frequency between them. And the tones are assembled in a triplet format. You play the tone, tone A, then comes tone B, then comes tone A. Then there's a silence gap for kind of short, like 200 milliseconds, and then it starts again in the same pattern, the A, B, A. So that's what this kind of unit, which in our case was 600 milliseconds long, is the so-called triplet. And the stimulus itself, it's a sequence of those triplets. Basically here we had now I'm trying to remember, 500 triplets together, which made, in terms of time, a long single trial played to the participant. That's, I wrote it here, 300 seconds, that's 5 minutes. So during those 5 minutes, because the subject is, the participant is exposed to this stimulus, they're going to start to report either what we call it as one stream and integration percept. This is like a galloping ta-da-da, 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 ta-da-da. That's one type of percept that they report. And then for some reason, the two tones, the sequences kind of separates into two parallel streams, which we call it here two-stream or segregation. So this is more like a Morse signal type of Morse code type of percept when they report like a ta, 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 there's a A or a B, 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 but they're kind of separated one from each other. So these are the two percepts under discussion, and the patient or the subject, the participant, will press a button reporting when they hear integration or when they hear segregation and back. So I have here an example of stimulus just to show what do I mean by what do I say, integration and segregation, but you know, I have to warn you, actually, I'm not sure if you're gonna hear it. Sometimes Zoom doesn't filter, and I think I forgot to select in Zoom. Let me know if you hear it. It's a little bit annoying, but we'll see. Do you hear anything?
I don't hear anything.
You do or you don't?
I do not hear.
Oh, okay. Sorry about that. Yeah, I remember that I was supposed to do something in Zoom. Anyway, we'll just go further. Just imagine that you hear like a galloping, da-da-da, da-da-da, da-da-da. That's integration versus tup, tup, tup. That's, you know, segregation, like a Morse code. And okay, let's see. I'll just move to the next slide to explain what we look at here. So basically in these kind of experiments, there are two sources of concurrently recorded data. One is the subject reported button presses. So here in this talk, just for you to remember, red is what is always associated with segregation, the Morse code type of percept when you have the two tones separated in the way you hear them. And blue is what corresponds to the one stream on integration. That's the galloping type of rhythm. So whenever the subject reports a bottom, I color here with a bar. So basically that's a subject reported data. I show here only 75 seconds, but obviously the report goes on over the 5 minutes trial. And in the same time, when the subject reports perception, because we work here with epilepsy patients that are in the lab because of the surgery, they're going to undergo for epilepsy. They ran this kind of experiment. They ran other type of experiments in this in the lab in the neurosurgery department. So we also have access to neural data from their brain. So in this case, I'm going to show data from core auditory cortex. So basically, there are a number of contacts placed on the heterogynous posterior medial, which is the equivalent of core auditory cortex in monkey. It's area A1, but here we work with humans. So these are kind of the type of recordings that are obtained from, I show you here 4 channels. Depending on the patient, we have 6 or 10 or 4. So I can show even for one patient we had just one recording, actually one channel recording from auditory cortex. So that's one characteristic of the data that we had to work with, and that actually justified mainly why we built these algorithms by bringing together 3 different ideas in the emerging methods in the literature, because there are some peculiarities of the data, and the most important one is the fact that we don't have access to many channels in the recording. It's a very low number, like I said, in terms of units, not tens or hundreds of recordings. So that's one thing that we have to take into account. Another thing is we're working here because it's perception and it's changing, and you need time for the perception to build and change. We run this experiment for seconds, hundreds of seconds. So I mentioned here the experiment is 5 minutes long. So basically, the data in terms of time, it's very, very long. So most of the methods work with short trials, like 1 second, for example, or 2-second trial, but while having many, many channels to record from. And that's, for example, the DMD, which is going to be a part of the algorithm I'll present here. Anyway, DMD is dynamical decomposition, where you can write these big matrices and look at the data. In this case, well, that's not what we have here. It's the opposite. It's a low number, very low number of channels compared to the time scale of the data. And we also, I should say, another thing that it doesn't work, let's say, with classifiers, because, I mean, if you take into account the 5 minutes recording, because we do not have many trials here as well. So for classifier, you need to run it. Usually you need a repetition of the experiment many, many times, and then, you know, separate the training data and the testing data, and then you run this algorithm. But that's not what we have, and it's not possible here, because that's just basically one single trial that we try to get the dynamics out of it. That's another thing. And I will also say, because we're looking at bistable perception, which is changing, it's kind of an intrinsic dynamics here during the 5 minutes. Even if you have many, many trials, it's not going to be very useful, because the same patient, the same subject participant, if you run the same stimulus next day or the next day, or I don't know, later in an hour, the perception is changing in a very irregular way. So you cannot have the button presses at exactly the same, let's say, 1.5 minutes in each trial. They're going to vary a lot. So even if you try to use the data from different trials in that sense, it's not going to pick up, for example, transitions from one percept to another, or dynamics during one percept, because in average, they're going to look different. So I hope that, you know, I kind of explain why we needed a different type of method to analyze the data. So the idea is, since we have these two types of recordings, the neural data and the behavioral data, we would ideally would like to see if we can extract what it's called a feature, right? A neural feature, something that we can extract from the data that potentially shows ups and downs which are correlated with behavior. So I'm just showing here an extracted feature where I have during the, let's say, one stream is up, but goes down during the 2 streams, goes up again in 1 stream, and so on. So like you see in a relaxation oscillator, typical in models, or like in competition models, when one is down and one is up of the activity, and so on, but looking only at the data. So I should just say that analysis of this type of data were done before, and but the typical approach, again, because we use statistical method, is to have the long 5-minute triplet when you have the sequences of repetition of triplets, and chop the signal based on the triplet. So that's what we call triplet, you know, base epoch. So 600 milliseconds, you just chop the signal, the recording signal in this way. So you create more trials. So in that case, let's say if I have 500 triplets in my sequence, I'll have 500 trials. Now I'm going to call them trials, but it's basically the same one 5-minute long trial, single trial that I mentioned before, and then try to run an analysis like in typical statistics, looking for difference in the average local field potential, and we did see some differences. That's type of the trajectory when you average across triplets which are in the red group, I mean two stream, or in the blue group, which is the 1 stream, and we saw some differences around the second A tone or the B tone, which were statistically significant. So yes, we've run this kind of analysis. There are other groups like Alex Billing and other collaborators that try to run this triplet-based classifier. Again, once you have many triplets, you can start thinking in terms of classifier and try to find a best hyperplane to separate the data. So there are analysis of this kind of data, the problem is you're kind of constrained to the triplet. So you cannot talk about the long-term dynamics when many triplets are associated with one percept before the switch to the next percept. So we wanted to go beyond this and actually look at the long-term dynamics in that. Why was that? Because I'm also working with competition models and competitional principle for bistable perception, and there are different models trying to simulate this kind of behavior. And here I'm just presenting a figure from Rankin and Rinzel, that look at auditory perception under triplet streaming, and came up with a neural-based model when they have three nodes, one associated with the A tone, one associated with the B tone, and another one associated with the triplet ABA, kind of the integration percept. And basically they simulate the dynamics in the model and show they were able to capture dynamics that shows, for example, the blue curve here, trajectories associated with the integration, the one stream, but then it's changing, and either green or red is going to be up, which means it's either the B tone only or the A tone only, which is heard. So that's a segregation percent and so on. So they define the one stream, two stream thing, and they compare the experiment, the psychophysic behavioral experiment, the durations, the dominant durations, which can be fit with a gamma or a log normal distribution, with what they could get from their model. And it was a pretty nice comparison. And they also show that depending on the difference between the tones, that you can capture kind of bias the perception to be towards more like the one stream or the two streams. But if you are in the ambiguous domain, you'll have this perceptual bias stability. So we try to see if we can connect the data, the real data, with computational principles behind perceptual bias stability, and for example, with this model of Rankin and Rinzel. So that was the goal, to see if we can extract from the data the long-term 5 minutes data when the patient keeps reporting their percept to features seen in computational models for perceptual bias stability. So we came up with an algorithm, but before doing this, I'm gonna just show I think it's a very simple example that explain the idea of the data, because in neural data, you don't have access to all variables in your dynamical system. So I think that's I'll start with this dynamic when you have a double pendulum, where obviously you have four variables in the dynamical system, in the ODE system, that will describe the trajectory. And the red and the blue dot are the points of the pendulum, which are connected together. So if you have the phase and you have the momentum and you generate, they're going to have a very complicated dynamics, which I'm simulated here in time. So if you look at the trajectory of, let's say, the blue dot only, you could see that because of the real dynamics, that's a complicated position in the plane, I would say in the space actually, for the blue dot. But what's important here is, let's say I'm the ladybug here, and I'm just gonna be able to monitor only the projection on the horizontal axis. So I'm the green dot actually, trying to follow, I said the blue dot, actually I should say the red dot here. So if I'm following only that projection, basically the trajectory I see, it's kind of an oscillation, yes, with some irregularity, but it definitely doesn't compare with the complicated trajectories seen there. So basically what I'm trying to say is, while your real dynamical system generating the trajectory might be very complicated, and trajectory can be very complicated, because you can observe only certain things from a finite number of things from the trajectory, maybe a projection, maybe on a plane or on a direction, you are not able to see the real dynamics, the real trajectory. The question is, can I use this to infer something about the original trajectory? So that would be nice to know. So in a similar idea, now the system, the dynamical system is the brain, which has a lot of activity. Obviously, there, if at least if you take the Hodgkin-Huxley model, you think about many differential equations describing many neurons which are connected, and then you have synapses, and you come up with those equations. So it's thousands and thousands of equations if you would think in terms of the dynamics of the brain. Do we have access to this dynamical system, which here I just say x dot equals f of x? No, we don't. What we do have is access to some measurements, for example, of a number of contacts here that I color. So there are just some measurements which are not even maybe x1 or x5 or some of my original variables, might be a transformation of those, like a function of x1 or a function of x2 that I'm actually recording, and that's the representation from different contacts of an LFP recording. So so the idea is how can I relate this data with an analysis? So if we imagine that the dynamical system represents the trajectories and x dot equal f of x, I am working a space. Basically, I don't know here the governing equations. I don't even know probably the dimension of the space, the original space. I imagine that, okay, maybe the trajectories do not follow the entire Rn, but stay on a manifold, a lower dimensional manifold, some dimension for this dynamical system, the trajectory of interest. But again, I hypothesize this is probably the case, but I don't have information about this manifold either. And again, a nice example would be like in Lorentz system, when you have these two attractors, that's a three-dimensional. We know that it's going to be chaotic there. Well, the manifold is kind of two-dimensional compared to the three-dimensional space. So that would represent like a manifold, but maybe I don't have information about this one. The trajectory itself, x, may be transferred from using some function, some observations on this manifold that I called here g function, might transform x into some g of x. I don't know the observation function itself, g, but what I know is the image of it. So that's what I will know. That's a scalar measurement. That's what I would say. This will give me for Y1, I'll have the recording from one channel. For Y2, that means recording from the second channel. So, I have R channels in my recordings, and these are the observations. So the question is, can I infer anything from Y about the original dynamical system to capture what's essential in, let's say, during one experiment, about the separation in a dynamics, which will be correlated with perception. So I mentioned that there are different methods that people implement in order to look for data-driven type of dynamics. And one is called time delay coordinates, another one is diffusion map, another one is dynamic mode decomposition, actually with a new format, extended dynamic mode decomposition mode, that depending on the type of data they are applied to, they were quite successful, independent on each other. Because of the peculiarity of the data that we have, and I mentioned before, low number of channels recording, however, very long recordings in terms of time, no access to repetition of trials. Also the fact that because of the perceptual bistability in behavior, we hypothesize that the data itself is stay on a manifold which contains two attractors, which will be associated with the two percept. We were not able to obtain much by implementing each of those methods separately, but we realized that each has an advantage that if you pull them together may be successful, and that's what we've done. We put together those three algorithms, ran the data, the intracranial recordings through the time delay coordinates, and then use that result in a diffusion map, and then in the extended mode decomposition to basically extract eigenvalues, eigenmodes, eigenfunctions that were used to project lower dimensional representation of the data, and use some of those eigenfunctions to actually show that if you go along those eigenfunctions, you will see correlation with the two percepts. So basically, we were able to construct those attractors associated with the dynamics, the neural dynamics, which correlated with behavior. So I'll try to explain each of them a little bit shortly before I show the results, because I think that's important. So for time delay coordinates, again, for simplicity, I use an example from the Lorentz attractor when we have 3 variables, but let's say you can observe only one of them. So I represent it here, x2. So time delay coordinate means we're gonna take snapshots of the same trajectory, but shifted. So that's the delay, and shift it again and shift it again if you have many delays. In this case, I just gonna take one delay, and I'm gonna look at the original trajectory I observed, so only x2, and its shifted copy with some delay tau. Now, if I represent those two components in like a plane, and I'm gonna represent the points, you see that, well, you can extract a structure which is pretty similar maybe with what is the original trajectory. It's not perfect, and we cannot expect to have perfect, but you can learn maybe about this separation in the attractors by just looking at the delay coordinates. So that's related to this is the Taken's theorem, which says, well, if you have a trajectory which evolves along a dimensional manifold, lower dimensional manifold, and you have those nice enough observations, so not the trajectory itself you see it, but some image of that g of x, and you take some copies of the trajectory, so that not the trajectory, the observation of the trajectory. So the y of t, you take that, you shift it and take a copy of it, then you shift it one more time, so it's like t minus 2 tau, and you shift it several times, let's say s. So you have a number of copies just shifted, and you pull them together. Now, if you make enough shifts, enough copies of these trajectories, and that's let's say the dimension of y times how many copy you have here, so that's s plus 1. Now, if this number is large enough and they compare to 2 times the dimension of the manifold, you'll try to reconstruct. And in some cases, you can relax actually this constraint, don't need this inequality. Then the newly constructed trajectory will yield an embedding of the manifold itself. So you may be able, through this methodology, to actually construct points on the manifold. They are not connected as a trajectory. It's just like points staying on the surface, or more general, a manifold of it. So in terms of LFP, the way we have the ECOG data, if you have a bunch of contacts, channels that you record from, now at each time point, you can create a vector by taking the values for all those contacts, and that's what I've done here. So at time, let's say tk, I'm going to read the values as points in a space, like we use t, time, as a parameter, right? To parameterize, like in the dynamical system, create this vector, and then this is for a time point, and then I will take into account the delays of it with 1 step or 2 steps or 3 steps or s steps. So this is the number of delay I'm going to take into account. And due to some theory developed for time delay coordinates, it looks like for data it's advantageous to give less and less influence of the points which are longer, behind, much more behind in the past. So it's use an exponential which with the delay itself here to reconstruct this vector. Now, then you assemble those together to construct a big matrix and try to look at the dynamics of those points. So this basically what we're doing, we transform the points, original points, in much higher dimensional space. So it's like taking the points, the real points that you have from the recordings, and moving in. I'll just say, I'm going to go from the Earth to the Moon. So I just have huge number of points there. Now, what we've done, we actually increased a lot the dimension of the points from the recording. So you would like to have an analysis of those points. So it's better if you can go and get a low dimensional projection of those. So that's the idea with the diffusion maps. In the diffusion maps, basically, I'll take those large, very large now dimension of points that I created, and I'm going to try to compare everybody with everybody to see if it's a distance. So that's usually you want to see the distance to create a neighborhood in this huge space, dimensional space between the points that we created. So to create a distance, you can use the Euclidean distance, but you would like to create neighborhoods which, points which are close together. You can see them. If they're further apart, you separate them better. So having a Gaussian type of separation will, emphasize the points which are close together and will just kick out the points which are relatively far away. So with this use, you can construct a matrix, and the matrix is going to be symmetric. So you can get the eigenvalues, eigenvectors of this matrix. That's what is called the graph Laplacian. Now, the eigenvalues will be real because we work with a symmetric matrix, and you have those eigenfunctions, which are features of the points of the data, and you can take only a lower number of these eigenfunctions to actually analyze and look at the data. Now, that's important because these are features extracted from the data itself. Can we learn a lot from those? Well, sort of, but it will be actually better if we can use them as what it's called a dictionary of functions, or the dictionary of features in the extended dynamical mode decomposition. I don't know if you're familiar with this one, but it does need a number of what they call a dictionary of function, like a basis, to try to project the trajectory of the system on this basis. And most of the time they pick sine and cosine and polynomials, for example, as dictionary of functions. What we've done here, we use actually eigenfunctions derived from the diffusion map of the data. So we call it data-driven actually functions, or to use. So the last part is like, with the Koopman operator based extended dynamical mode decomposition, is to what they're doing is assuming that you have a dynamical system and you have the trajectory given by this f t of x, which is a nonlinear, right, trajectory type of system, and you have a map which takes the trajectory in the real space or the complex space. The operator, I think it's better to show here, will actually consider this change of the trajectory itself in another copy where you actually use a linear operator, but it's an infinite dimensional one. So Koopman showed that by the use of this infinite dimensional operator, you can actually represent and explain nonlinear dynamics of a finite ODE system, for example. So there was a group of people that researchers that work on approximation of this linear infinite dimensional operator and through a finite matrix numerically, and by constructing this matrix using I just show in the previous thing that these are is the dictionary of functions extracted from the previous step in the algorithm. I can now look at the eigenfunction, eigenvalues, and eigenmodes of this matrix, and this will be complex, and that will represent the spectral features of the data that we are analyzing, where the time series in the eigenfunctions will be will show the series of the, let's say, 5 minutes, but now it's just some of the directions of the data that we extracted. Then the eigenvalues themselves, which are complex, will encode the frequency and the amplitude, but the amplitude usually not the amplitude, I'm sorry, the real part, it's typically close to 0. So the frequency, it's going to be the one which will play a role. And if you want to know, because we had different contexts, something about the differences at the trajectories at different channels, then those eigenmodes will help with the spatial coherence. So that's pretty much, the algorithm. Now, what were the results that we got by looking at the data? So now, we have this number of channels with a long recording, a 5-minute recording. I do not take into account the reports, the behavioral reports. I don't want to look at them for the moment. I'm just gonna push this data into the algorithm, being agnostic of the behavior. Only at the end, I'm gonna compare with behavior to see if there is any correlation. So taking this data to the time delay coordinates, diffusion map, and Kuhlmann operator, we were able to actually identify an interesting phenomenon here. So what was that? So I said that eigenvalues and eigenfunctions are important. So let's look at the eigenvalues that we got, which are complex. So for all of them, you can look at the real part and the imaginary part. Now, imaginary part over 2 pi will give you the frequency in hertz. And I have here two types of trials that we ran. One was called control, where we actually force, because of the choice of the tones, the subject, the participant, to hear one stream, then 2 streams, and 1 stream again, and 2 streams. So we call it control because we knew what is the bias there, and we wanted to see if the participant indeed responds without knowing, only listening to the sound, as we would assume that's as it is expected based on previous psychophysics experiments reports. And bistable is where we just basically had an ambiguous difference between the 2 tones A and B, and there were no change for the all 5 minutes. It was just the same triplet repeated again and again and again, to see if, and the participant reported changes in perception, but we want to see if there is anything in the data that will still capture this type of dynamics. So looking at the eigenvalues that we extracted from the data, we realized that in all cases, so these are different trials controlling this myself for the same subject, but I'm going to show you for different participants that we had. The eigenvalues are actually organized. You have complex eigenvalues, so they come in pairs. That's what you see here, 2 of them all the time, are organized on multiples of some particular frequency. And that's what the horizontal line shows here. So what's that particular frequency? Well, it turned out that it was actually 1.67 hertz, very close to the 1/0.6, and that 0.6 in seconds is exactly the length of the triplet. So basically, this is the frequency of the repetition of triplet in the sequence of the stimulus itself. So that was the first observation, and we thought that this was very important because it looked like somehow the frequency of the repetition, so the stimulus characteristic, was encoded in the data itself. So again, that was one observation. So we noticed that there were 2 different branches based on the energy. The energy is mostly related with the real part of the eigenvalue. Whenever I had the same complex, imaginary part, I will assign to the first branch the eigenvalue which had a larger real part, and to the second branch the one which will have lower real part. So that's the way we organize the first branch and the second branch. So and we noticed that if we look now at the eigenfunctions corresponding to those eigenvalues, if you stay on the first branch, well, there was pretty much like a simple cosine or sine that feature, and there was no difference, statistical difference between the points, between the 1 stream and 2 stream. The same here for the bistable. But looking at the second branch, for the same type of eigenfunction, there are some differences. So it's like a modulation of the first eigenfunction here. And the modulation, interesting, was actually given by the first eigenvalue on the second branch, which actually is real. So that was also a point of separation between the 2 branches. One of the eigenvalues becomes real, and that corresponding eigenfunction was actually showing the type of feature that we were looking for, in the sense that some are up and some are down, and there's a correlation with behavior. So this shows data from, like I said, one single subject in two different control and bistable long trials, but we did this for different subjects. So that's like 335, that's one of them. There's a different subject, a different one. So basically here I'm showing from 6 different subjects, and while the structure of the two branches might be different, there's variability from subject to subject, we still saw this consistently of having two branches of eigenvalues organized, and one associated with mostly with the stimulus itself, and the other one, which we call is most associated with the perception, because the second branch, especially the eigenvalue and eigenfunction corresponding to what you see here, green, was playing the role. So in the interest of time, I'm going to skip these slides. Basically, what I'm showing here, we're going to use that eigenfunction to actually make a prediction using the phase relative to that first eigenfunction on feature on the second branch to actually make a prediction about the button presses. So we don't only capture changes to say, okay, there's a change in the percept, but we also look at the short time scale, the timing of the button press. And I will try to actually show the geometry of those instead. So after after we got those features separated from the data, we try to see if there's any correlation with the percept. So basically what we use a 3D representation by taking into account the real eigenfunction. So that's kind of the trajectory in terms of time. And we took the first complex eigenfunction on the second branch, taking the real part and the imaginary part, and we plot those together. So when you plot those together, this is the example for one of the subjects, you get this 2 hourglass type of manifold, where the blue points are coming from trajectories on the associated with the one stream percept according to reports of the patients, and the red one to the two streams percept. And if you can also look at actually the A and B and A organization of the tones in each of the two parts of the hourglass, and they're pretty nicely overlapping at each trial, at each triplet, the points return to similar positions along the hourglass. And just to kind of wrap things up, I show here the representation for different subjects. So these are the 6 subjects I show you in a previous slide, where basically the features, the representation, 3D representation of the 3 extracted features shows similar type of manifold representation, this kind of separation in 2 parts of the hourglass, when one is associated with one percept and the other with the 2 stream percept. Well, they're going to look different here and there because there's more variability or more noise in the data, but pretty much the separation can be done in the space of those features. We can use the eigenmodes from the Koopman operated development to go back and try to reconstruct the data in terms of the original channels. Channel 1, 2, and 3 that are used here for the representation. And you can see that, well, that are much more differences between subjects now, if I'm going to look at the data in the channel space. And definitely if we'll try to find a hyperplane or another way to separate with classical method the data, you can see there's really a low chance to do it, because there's no really hyperplane to do it. It's much more nonlinear and much more difficult to organize. But if you look in the feature, it is the way it is. So I'll skip this part. I wanted to connect with a little bit with the modeling, but I'll just say that what we've done, apply these dynamic data techniques to learn features about trajectories of the real data, neural data, and try to bridge this analysis with classical theory from dynamical system. And with these applied techniques are actually wild settings with temporally structured data. So we think that the algorithm can be used in many more cases. Thank you. Sorry for taking more then.
No problem. Well, thank you very much for your talk. Do you have any questions from the audience?
I can jump in with a question, and I apologize if you talked about this. So you showed us kind of 6 different subjects, but how many subjects did you have?
They're actually the 6 subjects that we had here.
Okay.
So it's not, yeah, it's not easy to get these epilepsy subjects. But yes, we did have this 6. Oh, we originally had 10, I believe. But we had a previous paper when we look at the data for all the subjects and try to see through the classifier comparing the triplets if there is any separation, and only 6 showed relative, I guess, about 70% separation. So that's why we focus only on those.
Okay, interesting.
But yeah.
So you've used both embedding and Kuban methods here.
Mm-hmm.
Are you familiar with Joel Rosenfeld's work?
The name sounds familiar.
He was a graduate student here. He's a faculty at some Florida.
Oh, I think it's is doing something for control system. Maybe it's like a modification. Is it that?
I think so he does some of that, but he also does a bit of Koopman operator theory. And my impression is that his approach is more general and potentially more robust, because he's, instead of doing the Koopman method and getting an operator that may or may not be compact, I think my impression, my very vague understanding is that he tries to find the best operator that is compact that can reproduce the data. And so I don't know if that's relevant, but
I think I've seen recently some of his work, and what drew me the attention is I think it's like a DMD for control system. So I think that may be relevant, and it maybe might work even better for us, because I did present this like this is like a autonomous dynamical system, right? The theory with Taken's theorem, but basically, if I'm thinking of the type of experiment I have, this is a forced system, because it's actually this auditory input which keeps coming, right? So that will probably be better projected in his theory, where he describes a DMD model for a dynamical system which is forced and controlled somehow. So yeah, I'd be curious to see if there is an improvement using, instead of probably the extended DMD, I would just think that the last step, I don't know how he calls this, his DMD, but now I remember that I read about his method. Maybe Liouville method or something.
Yeah, that's right.
Yeah. So to read a little bit more to see if implement that type instead of the extending DMD might actually be even more clarifying. But yeah, that would be interesting.
I have a couple more questions. So with regards to noise, I mean, does the embedding, I mean, how do you account for all the noise in the data between the embedding and the Koopman method?
Yeah, so okay, I relied a lot on what there's researchers, right? I mentioned for each of the algorithm, there are groups of researchers working on claiming. So the use of the diffusion maps was not only for having a lower dimensional projection of the data than used from was also because they were able to show, I believe theoretically, that by using that how's it called, Laplacian, graph Laplacian, that actually is a good method to control for the noise in the data. So it's because you're using more like a stochastic, that you end up with like a stochastic matrix, and through the projection of it, you are able to reduce the influence of the noise. Now, I cannot say at what extent we were able to do it, but I felt that was sufficiently good because we were able to find structure still in 6 different patients with really noisy data when you look at local field potential. So
yeah, it's
my
quite impressive.
expectation is that that method having in the middle help.
Oh, that's excellent.
diffusion.
And then for EDMD, did you decide on your own set of basis functions, or how did you come up with them?
Yeah, so for EDMD, you need to come up with a basis, basis for the functions. And we look at the literature. I don't know if you're familiar also with Cindy, so they use polynomials or sine and cosines. So we wanted to stay away from that. I mean, that's kind of a choice, kind of at random, I would say, in terms of. So we wanted to go away from that and get a basis of functions still related to the data structure. So again, that's why we applied the time delay correlate diffusion before, to extract this dictionary or basis of functions also based on the data. So kind of, use that one and then clean it up by using Koopman operator, I mean the dynamic decomposition. So everything is based on data instead of taking a priori, I mean, taking some assumption.
That's amazing. That's really nice. Yeah, takes care of kind of two of some of the biggest kind of shortcomings with the Koopman method.
Yes.
Which is Oh wow.
That's
Okay.
what we felt. That was
Excellent.
nice. Yeah.
Okay. Well, I guess we should end here. So thank you again so much, Rodica, for this really nice talk. It was really nice having you.
Thank you. Thanks for your invitation. Have a good day.
Have a good day, everyone.